gene.
Yeah.
Right?
Forgot
And
at the last minute.
yeah, and each of these sort of bright pink areas here are areas that are dedicated to the learning and production of bird song. That's the only thing that they do. So if you can kind of appreciate here that this is a fair amount of neural real estate in the bird brain. So these songbirds really care about the song that they're producing. They're devoting a fair amount of their brain to getting it just right. And what we've learned in recent years, and from my work and from other people's work, other lab work, is really what are these neurons here? So right now they just look sort of like nice pink little blobs here, but these are really complicated structures. You've probably learned kind of in past lectures here that these are cells that have different shapes. They have different molecular signatures, meaning different types of genes they express. They talk to each other in different ways, meaning that they synapse onto in different ways, and they have different electrical patterns of activity. So one of the challenges in neuroscience broadly is connecting brain and behavior. So how does the properties of the brain connect to different aspects of the behavior? And this is an enormously medically relevant question, because one of the major challenges today is understanding the molecular basis of, say, neurodevelopmental disorders, what happens in the brain at a molecular level, cellular level, that leads to deficits in behavior. And that's kind of one of the major sort of medical framings of my research. Okay, but I'm also going to talk about a fair amount in this lecture about even a bigger picture question. It's about how do you these amazing behaviors evolve. So I'd like to start with kind of zooming out a bit and looking at behaviors across the animal kingdom. So I'm giving a few different examples here of behaviors or different organisms that have pretty remarkable behaviors that are really spread across the animal kingdom. So octopuses, especially in recent years, have been known to be incredibly smart animals and have really complex repertoires of behavioral repertoires. Spiders build these incredible webs. Not all spiders, but many spiders build these incredible webs, but they actually have quite small brains, but they produce these intricate patterns. This is a species of cichlid fishes that have these really complex social interactions. So by social interactions, you can imagine they have to respond to what another organism is doing and then adjust their behavior appropriately. Again, a very complex behavior. Then in humans, you know, we can do amazing things. We play the piano. So I don't need to kind of remind anyone about the remarkable capabilities of human beings. But one of the most amazing things is that the brains of all of these different organisms look remarkably different. So this is looking over at cephalopods. They have these large sort of lobes on the side here, but they also have distribution of a lot of their nerve centers are in their tentacles. The spider brain has these sort of these central ganglia here, central regions that also have these offshoots which connect to different parts of their limbs. The fish brain, we're getting a little bit closer to us. It has similar parts of the brain that can be mapped onto the human brain. And of course, the human brain with its sort of characteristic folds over here. So these remarkable complex behaviors, all generated by different brain structures. So the question is, has evolution arrived at different neural solutions to generate all these different complex behaviors, which are quite a lot of similarities? So I think the best way to really get at this question is understanding both evolution and development. So how do these brains develop? And how have these developmental processes changed over evolution to give rise to these different brains? And so to zoom in even further, we can look at this idea of cell types. So different types of cells. So for instance, in your liver, you have hepatocytes, fibroblasts in your skin. These are all different types of cells which have a single type of function or of a certain type of function. In the brain, there's an enormous array of different cell types, all of which are important for the function of the brain. So we can look at how these cell types evolve over time, how they've changed across different organisms. And we can also look at how they connect to each other. So there's a sort of jargony term called neural circuits. So what we mean by that is basically how these different neurons connect to each other and influence the properties of their neighboring neurons. So as you know, these sort of connections between neurons is also immensely important for the function of the brain. So this is also something that we're interested in understanding of how it evolved. Okay, so going back to birds a little bit, birds and mammals. So there's this idea of convergent evolution. What that means is that you have two separated sets of animals or any organism, and they've evolved along different trajectories. But by some trick of evolution, they've come up with the same properties, maybe the same behavior, the same wing structure. So classic example are the wings of birds and bats. These are very different organisms that have separate evolutionary lineages, but they both independently come up with the ability to fly, and that's through the structure of their wings. So this is an example of convergent evolution. This has also happened with complex cognition and behavior. So over here, I'm showing a little, this is called a phylogenetic tree, just showing the relationships between these different groups of animals. And birds and mammals, particularly, have multiple instances of convergent evolution and complex cognition and behavior. So some classic examples. You have these western scrub jays. These are all around outside. Many of you probably have seen these guys. These are excellent food cachers. They will cache their food, nuts, seeds, in particular locations in the forest, and have amazing memory, spatial and temporal memory of where and when they buried those seeds. Squirrels also have really comparable abilities to do this sort of food cache. Crows, or corvids generally speaking, are incredibly smart. And I've been in certain cognitive assays do as well as other non-human primates on certain ideas of skills as in terms of memory, and also some evidence they have something like a theory of mind, understanding that other organisms have something like a mind or their own subjective view. And then finally, what I study are the amazing vocal repertoires like this, or vocal singers like this European robin over here, and songbirds in our own species like the wonderful Dolly Parton. I'm from Tennessee. I grew up about 30 minutes from Dollywood. We had season passes. She's my personal saint. Right, so there's this incredible array of different behaviors. And as you can see, birds and mammals, they're separated here. They diverged a long time ago, about 320 million years ago, well within the time of the dinosaurs. But yet they've somehow independently evolved these different abilities, or that are actually quite similar to each other. So how did that happen? Okay, so first I'm going to dive in and just look at the structure of the brain between these two organisms. And we're going to get into a little bit of jargon. I apologize. In mammals, you have these, this is kind of looking at the brain. If you cut my brain like this, kind of looking at it face on, it would roughly look like this. This is a mouse brain, so hopefully my brain will look a little bit more complicated, but for demonstration purposes, this is a mouse brain. This is a top structure, which is called a pallium, and this bottom structure called a subpallium. Pallium means crest, and so it's like this top area, which is sort of covering the brain. The bird brain also has a pallium and a subpallium. In mammals, this pallium is mostly made up of the neocortex. So this is kind of what we think about when we think about the brain. It's the areas that receive information, sensory information, areas for memory, for association, for learning, and also for controlling fine motor control skills. Those are all part of the pallium. But if you look at the neocortex of mammals, you have this beautiful layered structure here. In fact, you have 6 layers across the whole surface of the neocortex. This is really a hallmark of the mammalian brain across all mammals. If you look at the bird brain, they don't have anything like that. So they don't have these nice layers. Instead, they have this sort of collection of neurons which are scattered around in these kind of what we call nuclei. So even at this first level, the structure of the brains of mammals and birds really look quite different. And you can ignore this isn't jargon. Should have checked that out. But if you look at their individual neurons, they actually look pretty similar. Neurons, in particular, the ways they communicate with each other. If you dive in and look at how these different neuron groups, these different circles are basically just different neuron types. And again, this is a lot of detail, but I just want you to see that there's some patterns of the ways that these neurons connect to each other and communicate with other parts of the brain outside of this neocortex. Researchers for decades now have performed similar analyses on birds and found that even though they don't have this layered structure and they just have this collection of nuclei, the neurons are communicating with each other in a very similar architecture, very similar way. So you have these guys in the green here are receiving sensory information, both in mammals and birds. They both receive information from this other area down here, contacting these blue areas, and then they go to these red output neurons where they control movement and other functions. So there's this common pattern. So it brings to the question. So they look very different, but they have these similar patterns of ways of communicating with each other, neurons. So what elements of the brains of birds and mammals are what we call homologous? Yeah.
I missed the DVR.
Yes, and I threw it in here. Okay, I'll go back to that. I said to ignore it, but maybe I should not ignore it.
Now that I see what it is.
Yes. So that's a good question. So it's this dorsal ventricular ridge, which basically is like the bird's neocortex. And it's a really enlarged part of the bird brain. And as I'll show you, there's no equivalent to it, though, structurally in the mammalian brain. It's functionally very similar to the neocortex, but it's an entirely different part of the brain that the birds have used to basically gain their smarts. And that's part of what the work I'll show you in just a little bit too, demonstration. Okay, so what elements of the brains of birds and mammals are homologous? So, and I'll define homology in just a second. And what features evolve separately? So what was inherited from a common ancestor? And what did birds and mammals evolve kind of when they're going along their own paths? Okay, so what is meant by homology? And I think I have this slide here because I think it's a squishy term that people throw around. And so it's useful to kind of go back to what is meant by that. So homology was first defined by Richard Owen, famous paleontologist, British paleontologist, who defined homology as the same organ in different animals under every variety of form or function. So it's a little almost contradictory. This is talking about the same organ, but yet every variety of forms and functions. So it can be quite different. It can look quite different, but yet there has to be some core feature which connects that organ across different animals. And you can replace organ with any aspect of biology you care about. So brain area, cell type, gene, signaling pathway, these are all things that can be homologous across different animals. So there's a few features here that are key. So a homologous feature is similar across species. It's present in the last common ancestor, and it was continuously found along the lineage. So basically, some homologous feature has to, if you look back at the last ancestor of birds and mammals 320 million years ago, for something to be considered to be homologous, it has to be present in that organism. So a lot of what researchers do is try to infer what that looked like, what that last common ancestor looked like, by looking at extant organisms or by looking at genomic sequences and trying to infer what the genome of that last common ancestor. Okay, so currently there's kind of two ways of understanding brain evolution and this concept of homology. The first is what I'll call the cell type or circuit homology. All right, so in this model, we have these basic cell types here. So I showed you that more complicated diagram that had these basic structure here. We have neurons that take in input from these sensory areas. They contact these other sort of intermediate neurons, and then they contact these outputs. And the idea, amniote, is so birds, reptiles, and mammals are what are called amniotes, because there's an amniotic sac present. And that differs from amphibians, for instance. Amphibians are not amniotes. So the last amniote ancestor, the model is that it had all these neurons, this basic structure of these neurons and the way that they communicate. Then, as it evolved, as these different lineages evolve, these basic neuron types are present in the different brains here, but they're just sort of distributed in different areas. So in this way, we can say that these neurons are homologous. They've been inherited from this last common ancestor. But there's sort of a competing model, which is called field homology. And this model says, if you look back in the amniote ancestor, there's all these different domains here. You have medial, dorsal, lateral, ventral. You can basically think of these as just sort of like big sectors in the brain. The brain is divided up. And under this model, These are present, all these different domains are present in all existing animals, excuse me, all existing amniotes, but they've expanded in different ways. So I mentioned before that the pallium, particularly this area called the dorsal pallium, that's where the neocortex sits in mammals. But this other area of the bird brain and also the reptile brain has gotten much bigger and has become more like the neocortex. So under this model, you have that these areas, the bird brain and the mammalian brain, are really not homologous in terms of what they can do, because it's derived from these separate subdomains. And historically, there's been sort of like an international battle around these two models. The Americans have favored the cell type circuit homology, and the Spanish have favored the field homology, and there's been a lot of name calling in the literature. God, these people don't have any training in embryo development, blah, 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 evolution, or how could they possibly say? And so it's led to a lot of contentious emails.
Bradley, maybe I'm getting ahead of the game
Yeah.
here, but I know that one of the big things is having this picture now of all the neurons in
Yeah.
the Drosophila brain.
Yes.
And last year Dan Turner Evans talked to the
Yes.
group. So where would insects be on there? It's kind of having that great resource in Drosophila brain, does it actually help us with any of these things?
Yeah, I will get into, basically that, not necessarily comparing with insects, but comparing across like these groups here.
Mhm.
I don't know if anyone has actually directly compared insect neurons to these more, maybe they have, they've done that. There's been a remarkable number of comparisons, basically using these resources to put together a cell type tree of life, essentially. Yeah. Okay, so which model is correct? That's always the dicey term, correct. Are both valid in some respects. Probably you realize the way I framed it, the answer is yes, they're both valid in some ways. And how can modern molecular biology, that's the title of this course, assist to resolve these kind of big thorny questions? Okay, so a little detour. I'm going to go into what I study. And kind of argue that the organism that I study, songbirds, can be used to address these questions. All right, so a lot of my lab is dedicated to studying bird song to understand how the brain learns and performs motor skills. And I take an angle of development and evolution to kind of to understand how this, to get a better understanding at a molecular level of how this occurs. So there's about 5,000 species of songbirds in the world. So there's about half of all bird species. It's an incredibly specious group of birds. And remarkably, each of these birds has their own species typical song. And there's, as you probably know, just kind of walking around beautiful forest around here, especially now, the birds are going crazy right now. There's a tremendous array of different species that have really complicated. There's a couple features of why this is a really great model to understand motor skill learning, but also brain evolution. So first, this is a learned complex behavior. And so the baby songbirds don't come into the world knowing how to sing. They learn how to sing, basically by memorizing what they hear around them from other birds of the same species. And then they start babbling. They kind of start to do this, they practice over time through a period of sensory motor learning. And then eventually, about 3 months after they were born, they produce what's called we call it a crystallized song. Down here, this is a spectrogram. This is a way to visualize the sound. This is a time frequency plot here. And each of these little sort of units here is what we call a syllable or a note. And this is kind of like the fundamental unit of birdsong. We've labeled it with helpful little letters here. And hopefully I'll have a little snippet of a song of the bird that we study. Let's see if it works. Did you hear that? Okay.
Yeah.
So there's a couple of aspects of that. It's incredibly fast. They're producing these syllables on the order of each of these notes is about 30 milliseconds long. It's also very precise. So this bird will sing that song hundreds of times a day over the course of its life, and it will change very little. So it's a remarkably complex and well-learned behavior. And another feature which is great for what we study is that it has this dedicated neural circuitry, which I showed you in that image at the very beginning of my presentation. This is a little cartoon version of that. That shows all these different regions of the brain, and there's arrows showing how they're connected to each other. And these areas are dedicated to both the performance and learning of song. So what we can do is we know that, say, this area in red here is responsible for the pitch of song and the loudness of song. This area in orange is responsible for the ordering of the syllables and how fast the song goes. So it's an amazingly powerful system to be able to go, how are these neurons, how they're firing, change, how's it changing in response to different conditions, how is the molecular aspects of these different songs changing. It's a really great way to connect brain and behavior. And there are really strong analogies to the way that humans learn how to speak and the way that birds learn how to sing. I'll talk about one of them here. One of them is that both birds or songbirds and humans have these dedicated vocalization areas. So in songbirds, this is a different representation of what I just showed you, basically showing all these different areas in orange here, which are responsible for song. But of course, in humans, we also have dedicated speech areas that are not found in other primates that do not have complex vocalization. So some famous ones here you might have heard of are areas called Broca's area, lesions in Broca's area. Those individuals cannot produce coherent speech. You have areas like Wernicke's area over here. Those people can produce intelligible speech, but they can't understand speech. So they've lost the ability to comprehend speech, but they can hear other sounds perfectly fine. And so there are these dedicated areas to speech as well as in birdsong. So part of the motivation for studying this system is to better understand how you can generate complex vocalizations using these dedicated areas of the brain. Yes.
If the person who cannot understand speech, is that simply the inability to translate sound, or would they be able to understand wording?
What was the last part?
Could they understand speech if it were written?
Yes, they can understand speech written.
So
Yes,
simply for the sound.
absolutely. Great question. So if they can read, that's totally fine. It's just specifically being
Sound.
exactly. Mhm. Yep. And then getting at this, so going back to this evolutionary question, so how can we use this system to get to this idea of the relationship of the homologous relationships between birds and mammals? Okay, so here I'm showing you this little cartoon of the song system. I'm highlighting these two areas, HVC and RA. I'm not going to make you suffer through what these actually mean, but just for this HVC and RA. And these are part of what's called the song motor pathway. So if you lesion these parts, any of those parts, then the bird can't sing. And so we know that these are very important for the production of song. And what's interesting is that these areas in particular have been really likened to different aspects of the mammalian brain, particularly different aspects of the human cortex. And so going back to this DVR, I have DVR throughout here. I really should not skip that out. So DVR is part of this dorsal ventricular ridge. And these areas are part of the DVR, which is this area which is not similar to areas of the human brain, of the mammalian brain. So it seems like this is a non-homologous, non-related structure that these two areas are in compared to the mammalian brain. So there's been these two different models. This one over here, the layer is basically this cell type circuit model, where it says that these two different areas, HC and RA, correspond to different neurons which are present in different layers of the cortex. This field-based model, the Spanish model, says that no. This is all part of this DDR, it derives from this really lower part of the brain over here. And it's not at all similar to the neocortex, which in mammals is found in this DP, this green area up here. Okay. So we wanted to, so the questions kind of boil it down are cross species who functionally similar neural circuits contain similar neurons. And for our system, to what extent are these song control neurons homologous to similar circuits in the mammalian neocortex? And the approach we took to answer this question, molecular biology to the rescue, is an approach we call comparative cellular transcriptomics. And I'm gonna break down each of these elements here to kind of describe how we did this. But before breaking it down, essentially what we did is we were able to isolate individual neurons from the brains of birds, songbirds, and from mammals. and then calculate their molecular profiles, what genes they have on, what genes do they have off, and then directly compare the neurons from birds and mammals to get a better sense of how they're related to each other at a molecular level.
What's the meaning of the axes there?
Yeah, so this is just a, so this means dorsal and posterior. So this is basically showing the relative orientation of the brain. So in this case, the dorsal, the beak would be over here for this guy, back of the brain over here. This is actually if you, a section if you're cutting the brain this way. So this is the front of the head would be out this way. Yeah, it's a different orientation.
No.
Lateral. Yeah, yeah. Okay, so what is transcriptomics? Maybe you guys have covered this or no?
No.
Okay.
No, you gotta explain that. one.
Great, okay. What is transcriptomics? Great. Okay, so these are really a suite of different ways to measure the expression of every gene in the genome. So in our genome, we have about 20,000, 25,000 genes. And each of our cells express some of those genes. And by express, I mean that gene is transcribed, turning it into RNA, and then often it's translated, turning it into protein. So some genes are expressed in some cell types, and some genes are not expressed in other cell types. And that pattern of which genes are turned on and turned off really dictates the function of those cells. So a liver cell will have many different genes turned on than, say, a neuron in my brain. And so transcriptomics broadly is a way to take a piece of tissue, say a piece of liver tissue, a piece of brain tissue, and ask, what is the expression level of all of these different genes? And so, for instance, I'll go through kind of a little diagram here. Say this is some sample of interest. You want to know what genes are turned on in these cells. So you can take the RNA out of these cells, you extract it, and then I'm going to zoom through a lot of complicated steps here, which you don't need to know. But basically, effectively, you use different complicated molecular biology to take that RNA and be able to count it. And such that you can say, well, I know that in this sample, I know that there were effectively 7 molecules of this gene. There were 15 molecules of this other gene. And so that gets you different levels of gene expression across the brain. So what can you do with that? So then you can take 2 different samples. Say I want to compare a normal cell population with a tumor cell population. And I want to understand what are the genes that might be underlying the tumorigenicity of the tumor population. So I can say what genes are different between those 2 different populations. And that can give me some insight into the molecular processes underlying tumorigenesis. So, and there's another, this is a hugely rich type of data. And so modern molecular biologists have been able to mine this data to do a whole suite of different comparisons between a range of different cell types.
So Bradley, you said what genes are different.
Yep.
And you mean which genes are expressed a
Yes.
lot and when
Yes,
they don't really
excuse
have
me.
different genes.
Yes, precisely. Yeah, good clarification. So essentially every cell in your body has the same genome. There's really not much variation. And so what makes the cells different is what genes are on and what genes are off, which genes are expressed and which genes are not expressed. So that's what I mean by, yes, exactly. Not the genes themselves are different, but gene expression. Now I'm going to make things even more complicated. I apologize, but there's been a recent real revolution in biology, where now you add on transcriptomics, but now you have single cell transcriptomics. And what that does, that allows us to not only ask how highly expressed a gene is, or how highly expressed or lowly expressed the genes across the entire genome are, but also in individual cells. So not just a big chunk of tissue, but if you could take a single cell and say what genes are it expressing, and then do that at bulk, basically doing 10,000, 100,000, millions of different cells, and that gives you an even much richer data set to understand the biology of your system. So what does this look like? So we have some tissue, organ, and the standard way that I talked to you about the transcriptomics is called bulk RNA sequencing, or RNA-seq. By bulk means we haven't separated out the individual cells. We just sliced all the cells, extracted their RNA. And what that gets you is you can look at if these different colors here, these bars are different genes. This tells you what is the average gene expression from, say, a diverse population of cells. So you don't know, for instance, how high was gene A expressed in this dark cell, or how high was it expressed in this white cell, which could be extremely important information, especially for tissues, which have a range of really a number of different cell types, which have different expression profiles. So there's been an innovation recently called single cell RNA-seq, scRNA-seq. Here, you separate all of your different cells into separate units, separate channels. And now you can ask, well, across these different cell types, how high or low is each gene expressed? And so what can you do with that type of information? So I'm giving you an example from breast cancer research, right? So here, what they're sort of, the authors of this study have, are drawing out here is a tumor, breast cancer tumor. And then looking specifically here, there's a range of different cell types that have different tumor properties, for instance, in yellow, blue, green. And with the single cell RNA sequencing, now they're able to separate all the blue, red, green, yellow cells, and then put it into this machine, which is called the DNA sequencer, to understand the gene expression across those individual cell types. Then you get a much richer picture of what's going on. So for instance, you can look at heterogeneity. What are the different cell types in this tumor? You can look at things like the tumor microenvironment, how the cells are interacting with each other. And then importantly, you can also do things like looking at the effects of different therapies. So if you provide a drug, you know, maybe an untested drug, which might be but promising for breast cancer research, you can apply that drug to a complex sample of tumor cells, do your single cell sequencing and ask, well, which cells were affected by this? What were the molecular profiles of the cells which were affected by this drug? Or you can look at how do different cells become resistant? This is a major topic in biomedical research now is drug resistance. So here you can look specifically at, well, I could apply this drug which used to work, and now there's this small population of cells which is really resistant to this drug. What genes are it expressing, which is different from other genes? So this is an example of how we can use these powerful techniques to really understand much more complicated biology. Okay, so let's go back to brains. So what can you do with brains? So you can identify distinct and possibly novel cell types, meaning like different types of neurons in the brain. You can determine how gene expression changes between different conditions. So for instance, how do certain neurons respond to disease-causing genetic mutations? This is a big area of neuroscience research. And then for this talk, we can compare how cell types differ between species to better understand how they evolved. Okay, so what did, and to kind of highlight this, the kind of how these technologies have really revolutionized our understanding of the brain. Here is a picture of, that was drawn by who's known as the father of neuroscience, modern neuroscience, Santiago Ramon y Cajal, who's Spanish. And this is an image from a part of the brain called the cerebellum. There's this beautiful sort of structure you can see of these neurons are quite complicated, but really, you know, we're identifying maybe a handful of different neuron types identified by their structure. Now, using these single cell RNA sequencing technologies, we have pictures like this, where this is basically a map of all the different cell types, and this is the mouse brain, but similar data sets have been generated for the human brain. And these authors had a little bit of an artistic streak and made this map, you know, similar to old maps showing sort of, you know, sailors back in the 17th century. But here we're saying these are all different neuron types here. It's hard to see, but these are basically a bunch of little dots all collected together. Each of these dots represents an individual cell that these researchers collected from the brain. And suffice it, obviously, a huge amount of complexity here. But basically, our understanding of how the brain is structured has become incredibly rich because essentially due to these technologies. And so now it's a really exciting time in neuroscience because of these approaches. Okay, so let's revisit this again, these central questions. So now we have these tools where we can do comparative cellular transcriptomics. So we can do these single cell transcriptomics, RNA sequencing, on a bunch of neurons from the brains of birds and the brains of mammals. We can ask, how are they similar and how are they different? So that's essentially what we did. This is work back from my postdoc that kind of led me here. What we did is we took the songbird brain, we dissected out these two areas that we care about, we think have similarities to different parts of the mammalian neocortex. So HUC and RA, we performed the cellular transcriptomic techniques, did some analysis. What you get out of it are these kind of funny looking plots, which are essentially ways to visualize the relationships between different cells in terms of how, oops. And so basically showing the relationships between these different cells in terms of how similar their gene expression is. These plots are incredibly difficult to understand. Even sort of people in my lab still don't really understand them. But they are nice because they take a lot of complex information and produce it in a way that you can actually visualize. So the main thing to take away from this is that we have these what are called clusters of different cells. So here, these are one different type of neuron, one type of neuron here in blue. This is a non-neuron type over here. This is a neuron type which makes myelin, which is basically the wiring of the brain. So essentially, we're using these technologies to understand the heterogeneity of these brain samples, to really be able to pull them apart. And now what we can do is we can compare each of these different types of cells to similar cells in the mouse brain. I'm going to zoom in on this a little bit further. Again, I don't want to focus too much on the details, but what we can do with this is we can find each of these different neuron types here. So we can uncover a lot of complexity in the brain. And I just want to highlight this here. So this is basically the lineage of different neurons that are being produced in the adult songbird brain. So songbirds were actually the first species, vertebrate species, that was discovered to produce new neurons in adult brains. So this is extremely controversial but before this period. And so there's some researchers in the 80s and early 90s that found that indeed songbirds are actually producing new neurons all the time throughout their entire life. And that motivated subsequent research in mammals and humans. There's much less adult neurogenesis in humans, and it's actually still quite controversial.
Sorry, but what is plotted versus what?
Yeah, so this is always the huge question where people are like, what are the axes here? And the axes here are meaningless. And this is one of the most frustrating parts of explaining these. Yeah, so this isn't a traditional plot where there's sort of, an x-axis and a y-axis. Instead, what's important is kind of the distance between each of these individual cells, and not so much how they're varying along these axes. So you can kind of infer from this, if you take, a little spot here and a little spot over here, the gene expression of those two spots, which represent neurons, are very similar to each other. But if you take one neuron, one dot over here, and then compare it to one that's way over here, the gene expression in those different neurons are going to be quite different. So it's just a way to try to reduce this really complicated information into a two-dimensional plot.
How many genes do you actually
Yes.
get RNA reads for in a?
Yes, exactly. Yeah, so we get about 15,000 genes. So you can think of this, for those of the data analysis inclined, each of these little dots here has 15,000 different points that are associated with it, or 15 different values that are associated with it. And so same across each of those. So basically, going this a little bit further, how this data actually looks, it's a big matrix, a big table that has the gene expression of all 15,000 genes in all of these different neurons. So that's immensely complicated. And if you were to plot that directly, you'd have a 15,000 dimensional plot, which is not good for mere human brains. And so people have developed different statistical techniques to collapse all that information down into 2 dimensions while still preserving the most important information or most critical information across those different cells. So that's essentially what you're saying.
Bradley, so when you did this transcriptomics, are you only looking at mRNAs, or are you looking at all these non-coding things that are coming up too now?
No, only mRNAs.
Okay.
So we don't look at these sort of new frontier in expression. We are planning on doing things like that using some of the technologies here, but not yet. Any questions about this?
15,000.
I was joking that there was a quiz after this, and, but no. Yes,
This may be a 30,000 question, but.
yes.
For the fact that the brains of the birds are so complex and are able to develop very complex
Mhm.
patterns of song and so forth, is there evidence that they are communicating
Mm.
with each other?
Mm.
And, I often see crows
Mhm.
gather in the afternoon
Yes,
and yammer away, fly away and everything like that. I always think they're coming together to discuss the stupidity of humans. That they
Yeah.
say.
right. Or ways to pick locks and, yeah, ways to.
But I mean, is there any evidence that that's?
Yeah. No, yeah, we definitely, I mean, the birds are absolutely communicating with each other. And so crows are technically songbirds, and they do have a song, but a lot of what you're hearing are calls, what we call calls. And those convey different types of information. You'll if you go out and you hear kind of a rattling sound, they do a beak rattle, and you hear it coming from the trees, and that's a warning sound or an aggressive signal. Often it might be because you are walking around. Yeah, but they have also just a range of their caws are also communicate different aspects of information. Interestingly, the song is, so we think of calls and bird song as separate things, and they're controlled by different parts of the brain. Bird song conveys not that much information, even though it's incredibly complicated. So essentially, what they're essentially saying are two things: either I love you or I hate you. And so they're either trying to attract a mate and trying to woo them, or they're defending their territory. An aggressive signal to say, stay away. And the complexity of that is in part thought to be akin to like the peacock's tail. It's a matter of sexual selection, where over time, certain species, certain females, and often in many songbirds, the males are the ones that sing, have selected for particularly complex songs over time, and they've gotten more and more complex over evolution. So yeah, so despite its complexity, there's relatively little information. Yeah. And I'm happy to talk about bird biology and ecology as well, in addition to all this completely abstract.
I have a question about, you've been talking about the distance between different cells
Yeah.
in the plot. How do you calculate the distance between
Yeah.
two types of cells?
You want the long answer or the short?
Well, you just start with the short.
Yeah. Yeah, so essentially, there's a first transformation that occurs in the data that tries to take it down to try to find the way in which the cells vary the most. So for instance, which genes are really changing the most across all of these different cells. And then you can do that for a number of different sets of genes. And then you basically calculate Euclidean distance between these different cells based on these genes that vary most. And so that's essentially kind of what we mean by distance. It's kind of the simplest way to think about it is you have 15,000 genes, and you can, for each cell, and you can subtract one from another. So, and then you can say, oh, well, if you subtract all the genes expression from one cell to another cell, you get basically zero. And so those cells must be pretty similar to each other because their genes are expressed pretty much at the same level.
Mhm.
But if you have something which is quite different and you get a larger value than zero, then that's essentially a larger distance, you can think of it. And so those cells are more different from each other. So that's kind of the root of what goes into generating these plots here. Yes,
What's the significance of the space between these colored dots?
you're asking the tough questions. Because it means something, but also potentially nothing. And what I mean by that is these plots are quite controversial in our field. because I can change how I do this analysis and completely change the space in between these. And so, people have even done something funny where it's basically you can say, yeah, I can make this look however I want. It actually does not, mean anything biologically. But what you often find, especially for things like these single cell RNA sequencing, is you find clusters of neurons and that group together. And so what we can infer from that is, at least within these clusters here, that these neurons are similar to each other. But I can't really take much information between, say, this cluster and that white space in between them, and say that they differ by X or some value. I can't really put a label on that. There's also other information. I mean, you can see that this is a line here, and that's not arbitrary. This is going from these adult neural stem cells all the way up into immature neurons, and going through all these transitional phases until it becomes a mature neuron. So these plots are tricky because space does mean something, especially when it's connecting actual points, but the space in between the points is relatively meaningless. I can, maybe I can, well, okay, go forward. But
Well,
go for it.
if you want to go on. Well, I'm trying to make a connection in an artificial neural network. The neurons are pretty much the same, but they get different weights when the network learns something.
Yes.
But if I understand you right, what you're saying here is that in a brain, in a biological brain, they're not all, these neurons are not all the same. It's much more complicated.
Yes.
They're all different.
Yep.
Is that a right way to look at it?
That is, it's precisely correct. Yeah. And to kind of break this up even more, I kind of skipped this, but when you brought neural networks, these neurons, when they fire, they excite other neurons. These neurons, when they fire, they inhibit other neurons. There's nothing like that in current artificial neural network design. And they'll do it in different ways too. Each of these excitatory neurons over here will kind of influence other neurons in this particular way. So there's a tremendous amount of complexity. And I'll say that there is an alternative to kind of standard neural network models called what are called neuromorphic neural networks, where they're trying to take principles of brain design and learn what lessons there are in terms of having these different types and different ways that they're able to influence the activity to generate other types of neural networks. Yeah, that's a good point. This is quite different. Okay, we're getting really into heady stuff. Okay, so is a cell type or field homology? And I'm going to skip a lot of work and kind of just show you some summary plots here, where we basically found that it was both. Maybe not surprising. What we found here, and I'm going to try to explain these in words, but we found if we look at these different areas of the brain, somber brain, NRA, they did correspond to different parts of the cortex. So it does seem like they correspond to different cell types. So this is very similar to this cell type homology model. If we looked at regionally though, this area in purple down here, which has the song regions in songbirds, was really actually quite similar to entirely different parts of the mammalian brain. In fact, they're most similar to the amygdala, which is a really important part for fear processing and emotional processing in general. So the story is that there's been these two different trajectories, and that the bird brain has evolved along a different way, and it's used this alternative area of the brain in order to generate these complex behaviors, which are typically associated in a different part of the mind. So mammals and birds diverged about 320 million years ago. In that time, the brains have really taken different evolutionary trajectories, resulting in brains with very different structures. However, they use similar components, so they have very similar neurons. So these are likely homologous neurons. So these are the things that we think are homologous, that have descended over time. But the different brain regions in birds and mammals converged during evolution. This perhaps explains the basis of the evolution of complex cognition and behavior in each group. And so that was kind of one of our takeaways from our study. Okay, so I'm going to zoom out a little bit and talk about kind of what we're interested in now in the last little bit of my talk. And this is going back to this idea of development and evolution of complex behavior. So everything I showed you now was all in adult animals. This is taking an adult, fully formed brain but there's a lot of power in understanding how you get to that brain. How did the neurons change over development across different organisms, and how have they varied or stayed the same? In fact, there's a whole field called evo-devo, the evolution of developmental mechanisms, which is thought to be kind of one of the core features of how is tinkering with different developmental processes is thought to be one of the main ways that organisms evolve. And so I want to kind of go back a little bit and talk about I think the power of the songbird system. So one of the addressing sort of open questions in that field. So one is this idea of song. So one thing that we don't know, and this is kind of a pet project, which no one will fund me for yet, but I'm trying my best, which is, I said there's about 5,000 different species of songbirds. They all have the different songs. Some sing incredibly complicated songs, and you also have things, songbirds like mockingbirds, which are incredible mimics. Then you have songbirds like the dark-eyed junco, who I just heard out just a little bit ago, who sing very boring songs and are quite simple. All of these birds have this same neural circuit, have the same structure. We have no idea how these are differing, how the circuit is changed or different across these different species that could allow some birds to sing very complex songs and some birds to sing very simple songs. Then being a little bit even wilder, there's some species of birds that have really remarkable dance. And so these are featured on all the nature documentaries because it's incredible to look at. I think of this as also kind of a different form of sort of complex learned behavior. And so one question we have is do they use essentially similar structures of the brain, but its output in a different way, in the way of sort of a dance modality, a dance behavior, as opposed to song. We understand these sorts of behaviors by understanding the circuitry. And the way we're doing it is through a process called comparative genomics. So this is a real strength of this campus, is taking the genomes of a bunch of different species and then saying what's different and what's similar across them, how have they evolved over time. Studying a process called gene regulation. Basically, this is trying to understand how one genes represented by the boxes here, kind of control the expression of other genes. How that some genes can make proteins, which then go back to the genome and either turn on or turn off other genes, which is an incredibly complex process, but is really important for development. And then again, getting this idea of cellular diversity. So it was noted here that neural networks have basically a single type of neuron, but in the brain, there's a huge amount of different neurons. And so trying to understand different aspects of how the cellular diversity evolved over time to be able to generate complex data. Okay, so I'll talk about two different aspects of what my lab is doing to kind of get at these big questions. So this is a little timeline of the important moments in a songbird's life. So there's egg laying, and while the bird is in the egg, new neurons are born. So neurogenesis occurs. And then, and as I noted before, neurogenesis essentially continues throughout the bird's life, although it reduces over time. After the bird hatches, about a month afterward, the brain becomes essentially receptive to memorize song. And so it will listen to songs around him. The bird will listen to songs around him, memorize it, and then it'll go through a period of song practice until it generates its mature song. So what we're doing is a couple different things. So we're looking really early in development for this process of neuron diversification. So basically, how do you generate new neurons in the brain? And how do you generate neurons that are able to produce complex behaviors like birdsong? The other aspect we're interested in is later, after the bird is hatched, and understanding motor skill development. So how are these brain regions that control song, how are they developing over time as this bird is going through its song learning? Basically, a process is very similar to speech learning. So at the risk of this is too, well, okay, let's go for it. So it's a big question of how do you evolve neural circuits? And neural circuits, again, are basically just different neurons that are connected to each other in different ways. And so there's this idea called duplication and divergence. And this is a real prominent way that new genes are evolved. So, and what happens with this, so you have a gene sitting in your genome, and when the DNA is copied, synthesized, or there can be errors. And sometimes genes get copied, duplicated. So you might have a particular gene that does some function, and during DNA synthesis, there is some error in that molecular process, which causes that gene, causes for them to be two copies of that gene. So now you have two genes that are doing the same thing essentially. But what happens over time is that there's less pressure to keep one gene the same, okay? Because you have this other copy that can do the thing that was originally doing. So over time, you can get mutations in that new gene, and it can acquire new functions. It can do new things in the cell. So that's one way that you're able to generate genomic diversity, how our genomes have gotten so complicated over time, measured 20,000, 25,000 genes. That number, a lot of that number is based on this process of duplication and divergence. So neuroscientists have taken this analogy and said maybe the same thing is happening to neurons in the way that they're structured. So this is a little tree, evolutionary tree. These are just showing different patterns of neural circuits here. And what people have argued is that this is some ancestral circuit here. This neuron connects to that neuron. At some point during evolution, there's a duplication of this circuit. And then, because there's two copies of it, now one of them can diverge and acquire new functions. There's some evidence that this has kind of happened in the brain. So if you look at speech control areas in the human brain, you find that there's a duplication of this area called the laryngeal motor cortex. So this is part of the brain which controls the larynx, so basically the vocal organ. And if you look in primates that do not have complex speech, there's this area kind of in the more ventral part of the cortex called the LMC, primate LMC. Humans have a human-specific LMC that are not present in non-human primates. And so there's this idea that in the human evolution of humans, there was a duplication and an eventual divergence of this motor cortical area. You can even look in primates. Some primates have really fantastic use of their hands, just like we do. Others do not. And the primates that have really complicated control of their hands, they have part of their brain duplicated that controls complex hand movements. So basically they have what's called a new motor cortex, and which sits alongside the old motor cortex, which are found in other primates that don't have complex use of their hands. So in songbirds, people have applied this same sort of logic, where you have in ancestral birds, like let's say a chicken. I like to rag on chickens because they're not very smart and they can't really sing. But they have basically the same brain architecture, where yellow region connects to this red region, yellow region connects to this green region. Songbirds, but in songbirds, you have layered on top of that this song-specific circuitry. So the idea is that there's been some duplication of patterns of neurons which have existed before, but they've been able to acquire new functions which have led to the evolution of song. Okay, so I think of this as a process of coordinated cellular evolution. So what we're trying to find out is what are the non-song, what we call sister cell types of song. So what are these cell types? So what are these pale cell types over here? And what makes them different from these song neurons? And then ultimately, what are the genetic mechanisms that drive the specialization or divergence of these song dedicated neurons? And so we're doing this for a couple of different approaches. So we can take finch embryos at different time points within the egg, and we can do these cellular transcriptomic studies where we can isolate their brains, produce one of these incomprehensible plots. And but here we can study how they're developing. So these are neural stem cells over here. We can see these little isthmi, how do you pronounce that word? Connecting these kind of islands of more mature neurons over here. So these are these excitatory neurons, these are inhibitory neurons. And what we can do is we can track the development of the neurons up until this point over here, and we think these guys in yellow over here are song neurons, which are just starting to develop, just starting to emerge from these other neurons. And then we're also studying this motor circuit maturation. And so I've talked about this a bit, but there are a lot of behavioral similarities between the way birds learn how to sing and the way humans learn how to speak. In particular, in humans, there's this sensory learning period where we're kind of listening to speech sounds around us. This can occur very early. It's not that even in utero, there are some sensitivity to sound. There's a sensory motor learning phase where babies start to babble. They start to kind of try out their vocal organs, and eventually, as I have two young children, they're right in this phase right now. They babble and they say, they say weird things, but they say weird things in a weird way. And that their phonation and their pitch and their tempo can often vary a lot between each time that they say it, but eventually they're able to produce really stable adult speech, much like all of us have, where we can say things very precisely and very quickly. Same transition occurs with songbirds, where they start to babble early in life until they are able to produce this extremely precise behavior, the song, which I had you guys listen to at the beginning. And I'll say this is even a more general process. Oh, excuse me, not that slide yet. So this is a little kind of snippet of what this looks like. So this is a 45-day-old songbird, and this is showing his song. This is the spectrographs. This is time versus frequency. And you can probably see that these syllables here, they look kind of noisy. So by noisy, meaning it's not very, there aren't these nice bars, these nice lines here. It kind of just looks like it's mush. And then over time, as the bird ages, there's more and more structure to the bird's song. You start to see that these syllables look a little bit more defined. And then finally, around 90 days, you have this really nice structure. The bird is singing what we call one motif, and then the second motif, they're essentially identical. So this process of going from variability or babbling down to stability. And this is a general process of developmental motor skills, generally speaking. So our ability to walk, our ability to kind of reach in a purposeful or grab things in a purposeful manner, they all go through this period of variability early in life. And then they go through, eventually they stabilize until we're very good at performing certain actions. So we want to know what's happening in the brains of songbirds from this process of going from variable, sort of babbling behavior to really stable, very well executed behavior. So one way we're doing this is we're tutoring birds in a computer setup, a computer tutoring setup here. This is a little finch sitting in a box, and we have a little microphone up here so we can record his song. And then we have a system set up here where he can hop on this perch and a song will play back. And over time, that bird will learn how to sing that song. And we can do a control where we have the bird not able to hear anything at all when he hops on the perch. So this is an isolation experiment. So we want to know how is the brain changing when he's not able to appropriately learn how to sing. So this is a complicated experiment diagram here, basically what we do is we transfer eggs to nests with two females because the females don't sing. So they're reared in isolation of song. And then we put them into two different conditions, either they're tutored or they're isolated. And then we perform RNA sequencing or transcriptomics on song and non-song regions. And then we can ask, won't go into this plot too much, but basically the more dots above the line here means that there's more differences in gene expression between two conditions. So here, this is looking at 60 days versus 40 days old. If we look in RA, which is one of these song regions, we see a huge number of differences. This area is changing a lot as this bird is learning how to sing. This is a non-song area just next to RA. Nothing's really happening. So there's a lot of specificity here. And then we can go in and say, okay, what types of genes are changing? As bird is learning how to sing, we see that there's more sort of cellular respiration occurring. It's becoming less, neurons are kind of stabilizing their shapes. And then we can also look at individual genes that basically try to understand better what is happening. But ultimately, what we think we can understand with this is really the molecular and cellular basis of developmental motor disorders, the dyspraxia and also specific language impairment. So this information, these are incredibly complicated diseases, and the molecular basis of these diseases is really not well known, in part because it's hard to study complex motor behaviors at a molecular level in humans. For obvious ethical reasons. But with the parallels to the development of song in songbirds, we can have a better sense of how these areas of the brain are changing to see if there's some core molecular features which might give rise to these differences. Okay, so I'll end with just kind of going, oh, go for it. It depends. Some birds, no. There's a famous experiment in our field where we took two different species, a song sparrow and a swamp sparrow. Song sparrows have very complicated songs. Swamp sparrows have very simple songs. You can teach a song sparrow a swamp sparrow song, kind of, but you can't, the swamp sparrow will never learn a song sparrow song.
So they have the structures that.
Yes, exactly. Yeah. But there are some birds that are, fantastic mimics. Again, like the mockingbirds, they will pick up anything. And I even have this book that I picked up at a library book sale that's like basically little flute songs that you could teach your canary to sing. But yeah, so there's a lot of, it depends on the species of the bird. All right, so I'll kind of end up here by kind of going back to these big questions of understanding the evolution and development of complex behaviors across species. And right now, neuroscience is really at this exciting time, in part because of all these modern molecular tools, which has opened up a huge range of different species to this sort of analysis. So maybe 10 years ago, really, if you wanted to do neuroscience, you had to study a handful of different organisms. You studied mice. This is a little bit of an exaggeration, but most of the organisms were mice, non-human primates, model organisms like Drosophila, fruit flies, and zebrafish. But now that you have, basically there's hundreds of different genomes which have been sequenced and assembled for a bunch of different species. Now we have techniques like single-cell RNA sequencing, where it doesn't matter what species you study. If the species has an interesting behavior and has an interesting sort of way in which it's controlling that behavior, you can study it, and you can have a better sense of the diversity of different behaviors and how what neural solutions have evolved over time to create these different components. So I think it's a very exciting time to really get at what are the core features that make brain and behavior possible. Okay, with that, I'll just end with a lot of the work here was contributed by my lab. We have a thriving research group over there in Sinsheimer. And we've been here for about 2.5 years, and so we're just getting started. Also affiliated with the Genomics Institute, which does a fantastic job of using a lot of these modern molecular techniques. And my old advisor, Michael Brainerd, and I'd be remiss to say that a lot of this work was funded by the federal government, and we are in dire times in terms of getting that money flowing and having basic support for basic research. So all your congressmen. Say it again?
Birds.
Yes, so parrots. So it's thought that in birds, the ability to imitate vocalizations has evolved 3 times, maybe twice, but so hummingbirds also can sing. Parrots and songbirds are the other 2 groups. So parrots are kind of a different case in that they don't sing per se in the way that songbirds sing, but they have incredibly flexible vocalization. And so they are even more fantastic mimics than, or more generally speaking, than songbirds are. So they are very much able to take what they hear and translate them into their own vocal production. And they have very similar brain structures to songbirds. In fact, there was a paper that one of my colleagues just published in Nature, and I learned about it because it was on NPR, where they looked at parakeets and did a direct comparison between parakeets and finches, and looked what's different, what's similar, not with this molecular stuff, but with kind of how their neurons are talking to each other. And it's very interesting. It's got a lot of similarities, but their parrots are doing something quite different. Was there?
When we call someone a bird brain.
It's not an insult.
If it is an insult, then we're actually
Yeah.
giving them too much credit.
Exactly.
It's good to know.
I mean, it's hard to, as I mentioned, chickens, I don't know. But and pigeons, but turkeys. Yeah. But songbirds and parrots especially are quite intelligent.
Chickens, I'm sorry. They're very intelligent.
Yeah.
I have raised chickens.
Yeah.
They are so stupid. But I think I answered my own question, is that of course we were the ones raising them.
Yeah.
So, they get stupid.
Yes. Yeah, I used to have chickens too, and yeah. Right, they were lovely, but they, yeah.
Have you got someone taking the word diversity out of all your applications?
Oh, I have worried about, I have thought about that, yeah, because that is something I talk about genetic diversity, cellular diversity. I talk about females,
Uh-oh,
female birds.
you're dead meat.
I know, there is a bit of that. I know. Yeah, yes, exactly.
That's very colored.
Plumage, yeah, exactly. This is, all joking aside, I mean, this is something that we are, very much worried about. And, people have already been, I had a member of my lab, a trainee, who's just getting started in his career, his funding was revoked. And so which is very disheartening. Obviously, for
NIH
him.
one.
And so there's things like this, which are starting to hit home. But yes, scrubbing, sanitizing for things like that.
Question.
Yeah.
Uh, Often the females don't sing. Do you know the basis for that?
Well, there's an interesting story actually behind that, where it's not actually true that the females don't sing, but many of the main model organisms that we study in neuroscience, that is the case, where only the males sing. Someone did an analysis recently, and they found that it's actually 70% of bird species, both the males and the females sing. But most of the sort of Western temperate birds, songbirds, there's a strong, what we call sexual dimorphism, difference between the sexes. There's some species of birds that do this incredible duetting between males and females, where the male will sing and then the female will sing, and it'll be no gap. It will be just this instantaneous trading off. And there was a fascinating study where they recorded the brains of the males and the females as they were singing to each other. And when so certain parts of the brain would light up, when it would be active when the one bird was singing, but even when the other bird was singing, it was the same pattern of activity. So their brains were essentially synchronized together, even though they weren't necessarily singing at that particular moment. So yeah, so there's a lot of interesting, but to get back at that, we do know that there's a burst of testosterone early around like the first month of the male bird's lives and for our species. That has a huge effect on the growth of these song areas and their ability to sing. But it's so that you can, in canaries, for instance, you can inject adult female canaries who don't typically sing. You can inject them with testosterone, and then within 2 weeks, they'll grow these brain areas and they'll start singing. So there's a tremendous amount of plasticity in their brains.
But it's real neuron different.
Yes, it's really, they look different. You can just, even by eye, they like.
Question.
So how are you doing this research? You go out in groups and do it, or it's all in the lab,
Yeah.
birds in cages?
What was the question?
How is the research done? Do we go out in the field and collect birds, or do we have a sort of a lab colony? Right now we have a lab colony. So we have a room that has a bunch of cages in there and a bunch of breeding cages, a bunch of nests in there where the males and females are mating. And then we have little sort of mini recording studios where we record their songs to see how they're changing. At some point, I would like to go out into the field, especially for this question of song diversity and kind of how brains differ across different species. But that's my background as a molecular bench scientist, so that part's scary to me. I've been talking to ecologists to do that stuff, and so maybe someday. Yes.
Interesting anecdote. The town in San Diego, the minor birds are not learn to mimic the song.
Yeah. Right. Yeah. So that they can maliciously learn different songs too, in order to annoy people around them. Yeah.
When you, in that experiment where you isolate a bird from song and it doesn't hear the song, is it then ever able later to learn songs, or is it just use that ability permanently?
Yeah, good question. So there's a period called a sensitive period or critical period, where the bird, it's within the first few months of life that the bird has to be exposed to the song. And that's the time that if they're not exposed to song during that time, then they won't learn the song. They produce something called an isolate song. So you can still tell that it's the same, it's the species, but it's not structured, it's much more variable. And so it's not, it really depends on the specific time. And so there's even tragic examples in humans where a child has been raised in isolation for speech, and they also have a really strong difficulty learning how to speak. Learning a second language is also a great example of this. You know, children that learn 2 languages, say when they're growing up, they speak both fluently. As we all know here, learning a second language after our critical period, if we ever learned a certain language, is incredibly challenging.
Have you ever tried taking one of these isolated birds that didn't learn to sing, and then pop a period of his life and give them a dose of testosterone?
Ooh, that's an interesting question. Yeah, so take an isolated bird and then and inject them with testosterone to see if it will kind of boost up their singing ability. What people have actually seen is that if you take a young bird who is in the process of learning his song, and if you inject it with testosterone kind of early, before it really has high levels, then the bird's song all of a sudden becomes super stable. Usually when they're practicing, he's like changing the pitch and the frequency and the tempo and all this stuff. You inject them with testosterone, all of a sudden it produces the same thing. So it becomes almost pre-stabilized. But I think if you did that for an isolate bird, it might kind of stabilize it a little bit, but it wouldn't be able to learn the song.
Oh,
yeah.
But you haven't tried it.
I haven't tried that. No. Yeah.
What's the research community? I mean, like I
Uh-huh.
did my work with Neurospora, a fungus, but there
Yeah.
are about 100 labs around the world that
Yeah.
do that. How many?
Yeah.
How many labs
Yeah,
are
I
there?
would say there's about 100 charitably, maybe more like 50 or 70.
You have meetings and
We have our songbird meetings and bird song meetings. And yeah, we all get together and talk about birdsong. And yeah, so it's a good community. And I think it touches on people like me, or there's people like me who do molecular stuff, people who do more behavioral ecology things. And so it's kind of a fun, group of people to get together. Yeah.
all right. Well, I think